here 35 years ago, but the words I had to look up to introduce her. Dr. Gabriela Cezar is, I'll read some of the description, again, so I don't screw up the words. She has identified biochemical markers and pathways that are altered by known disruptors of human development. For example, valproate, which I had to look up for you like me, that's epilepsy. But also alcohol problems, and that I do know something about being your county executive. She used metabolics of human embryonic stem cells and neural precursors derived from those cells. This approach offers a really unique advantage over other models since it employs cells derived directly from human embryos. Moreover, in addition to elucidating mechanisms of anti-epileptic and alcohol activity during early development, the biomarkers discovered in human embryonic stem cells are applicable to preclinical safety evaluation of pharmaceutical compounds and human development. You know, translating that to me, a county executive that deals with all the problems of alcohol in our community, we're the highest state in binge drinking, highest in underage drinking, highest in women's drinking, and highest in fetal alcohol syndrome. Her work will make a difference. Now, besides just that one example, she is a founder of a company called Stemina, and she is known not only, again, for her incredible science, but her incredible ability to translate her science to us. And so it is incredibly appropriate that she be conducting and leading here Stem Cells 101. With no further ado, a great honor to introduce Dr. Gabi Cezar.
What an honor, what a great introduction. Thank you so much, County Executive. Is there any way we could bring down the lights, just because I have some slides that have fluorescence? Okay, thank you. It's a pleasure, a great pleasure to meet you all. And I guess you can gather how important this World Stem Cell Summit is for Madison and for the whole global stem cell research community, right? So today we have basically scientists from every single country arriving in Madison. This is going to be the world epicenter. It's not just world in title. What this conference will involve is really a convergence of scientists, patients, policymakers, government representatives. We are just honored. Bernie, thank you once again. Bernie, I have to tell you a secret. Remember when I was driving you in the blizzard?
I'll never forget it.
I'm not a good snow driver, Bernie. I was just trying to keep you calm. I felt I always had to tell you that. We're going down the belt line. There are cars turned, police cars everywhere. This man is hanging on, and I'm like, don't worry, I know how to do this. I'm Brazilian, bro. So it's an honor for me to speak to you today. The Stem Cell 101 lecture is really a lecture that's designed to give you a comprehensive overview of what are stem cells, what are the major areas where stem cells can benefit patients, because sometimes you probably think, when you think about benefits of stem cell research, you think exclusively and primarily Parkinson's disease, spinal cord injury, right? Heart disease, diabetes. Those are indeed the first and foremost applications of human embryonic stem cell technology and adult stem cell technology. But I'm going to introduce to you other applications, such as the use of human embryonic stem cells and adult stem cells in the drug industry to prevent the fourth leading cause of death in the United States, which is severe adverse drug reactions. Did you know that? Severe adverse drug reactions are the fourth leading cause of death in the US. That's CDC data. And the reason for that is largely based on the fact that we have been making decisions about safety of drugs and chemicals to patients using exclusively animal models. So we make a decision whether a drug is safe for a patient based on what we learn in a mouse or in a rat or in a rabbit who are not going to take the drug. And so that's also an application of human embryonic stem cell research that I'm largely involved with. As County Executive mentioned, I'm going to show you further some of the research that we do in our lab, trying to understand the causes of neurodevelopmental disorders, particularly the effects of chemicals in the environment during human development and how that affects human development. So my background, I've been working in stem cell research and cell plasticity for 11 years. I had the fortune of working in the team that cloned Dolly the sheep. Okay, this picture's so outdated, but I had to show it to you. In Scotland. Sunlac, right? Scotland. I counted 45 days without seeing the sun, thus the paleness. Anyway, I was first exposed to stem cells, mouse embryonic stem cells, working with Ian Wilmot in his lab in Scotland. I did my PhD here in Wisconsin. And then I also worked at a company called Infigen, which we were cloning cattle and pigs to be able to generate organs, in the pig case, organs for xenotransplantation. And then after I got my PhD, I went to the pharmaceutical industry and worked at Pfizer, the largest pharmaceutical company in the world, for about 4 years, heading their stem cell group to apply and understand how best stem cells can benefit patients, not only through therapy, but also through creating better models to predict side effects. And then I left Pfizer 3 years ago. It was a very difficult decision. It's a great company to work for, but I had a drive, and really I was hungry to work on human embryonic stem cells. That's why I left the pharmaceutical industry and came here to Madison and started our laboratory. So my background is largely on embryonic stem cells in drug discovery and toxicology. Just some of the issues that we can address is, for example, in terms of preclinical safety, QT prolongation is a cardiotoxic effect of many drugs. And we're able to generate heart cells in the dish using embryonic stem cells, and then we expose them to drugs. This is just a readout of what a cardiotoxic compound would do. These are calcium waves inside heart cells, and the calcium waves correlate with contraction. So basically what this means, and you can see these cells beating faster in the dish in response to a toxic drug. We can better understand how drug metabolism works, and then we can discover new drugs for obesity, for example. These are fat cells. You can see they're called adipocytes. You can see the lipid droplet inclusions inside these cells. And we can, for example, discover new drugs for Parkinson's disease. So these green cells here are the specific type of neuron, of brain cell, that is damaged during the onset of Parkinson's disease, the dopaminergic neurons. So we can create these cells, and then in the cell culture dish, we can discover new drugs for Parkinson's disease, right? So not only could we use these dopaminergic neurons to potentially transplant these cells directly into the brains of patients with Parkinson's disease, but we could also use the cells to discover new drugs for Parkinson's disease. So UW-Madison, as you've heard, is, and I hope you knew that before you came here, it is a world leader in stem cell research. The first and foremost factor that determines that is that human embryonic stem cells were discovered here in Madison by Jamie Thompson, Dr. Thompson, who's on the cover of the Wisconsin State Journal today. There's a great article at the State Journal covering this summit. And I had the privilege, Jamie was part of my PhD committee, so when I got my PhD here at UW-Madison, Jamie was one of the members. Now, there are other very important